learners, welcome to JHL Zoology, Integrated Study of Animal Science for UG students. Today in this video lecture, let us know about Hybridoma technology, the production of monoclonal antibodies. Learning objectives of the topic are: the learner will be able to know about Hybridoma technology, to understand the concept behind the production of monoclonal antibodies, to know the preparation of hybridomas, and the application of monoclonal antibodies. Now let us know what is this hybridoma technology. So hybridoma is a cell, it is a hybrid cell obtained by the fusion of B lymphocyte with a tumor cell of an antibody forming system or a tumor cell of the B lymphocyte, which are called as myelomas. The hybrid cell thus formed possesses the ability of producing the antibodies due to its B lymphocytic genome and the capability of indefinite growth in vitro due to the tumor cell involved in the fusion. Therefore, specific hybridomas are either cultured in vitro or passed through the peritoneal cavity of the mouse to obtain the monoclonal antibodies. This technology is called as a hybridoma technology. Let us now see the discovery of the monoclonal antibody production. The monoclonal antibodies were produced by George Kohler, Caesar Milstein, and Niels Jern in the year 1975 by the hybridoma technology, for which they were awarded the Nobel Prize in 1984. Then let us see the principle behind the hybridoma technology. It is based upon the somatic cell hybridization, in which the fusion of antibody producing B lymphocyte with a myeloma cell will be done. The myeloma cell is a continuously dividing cell. So as a result of the fusion of B lymphocyte and the myeloma cell, a hybridoma cell is having both the characters of the parental cells. The hybridomas produced, they are capable of producing the desired antibody in large amounts. This large-scale production of monoclonal antibodies is called as hybridoma technology. Let us now know what are these monoclonal antibodies. Mono means one, and the clone is a strain of cells descended from the single cell. And the antibody is a molecule of animal origin that has immunological activity only against the antigen to which it was made. Means it is having the immunological activity against the antigen, a specific antigen for which it was made. There are different types of monoclonal antibodies depending upon the source. Here they are the murine source monoclonal antibodies. They are of the rodent origin. Chimeric monoclonal antibodies, they are the chimers combined with the human constant regions with the intact rodent variable regions. Whereas the last one is the humanized monoclonal antibodies. They contain only the CDRs, complementary determining regions of the rodent variable region grafted onto the human variable region frame. Let us now see the properties of the monoclonal antibodies. So, the first property is they are produced by the hybridoma cell line. Monoclonal antibodies are produced by the hybridoma cell line. They are the single type of antibodies that are produced by B cell clones of a single parent cell. They are directed against a specific epitope, a specific antigen or antigenic determinant. Monoclonal antibodies have same antigen binding site. Antigen binding site is same for all the monoclonal antibodies produced from a single parent cell. The potential cross-reactivity is very low between the monoclonal antibodies. Now let us see the steps involved in the production of monoclonal antibodies. The main steps involved in the production of monoclonal antibodies are immunization, the cell fusion, culture in the hat, and screening and culture. Now let us see all the steps one by one. So immunization. What is this immunization? Here you have to select an animal, the mice or the rabbit, and that is immunized with a desired antigen. The immunization is achieved by injecting the antigen along with a suitable adjuvant either subcutaneously or in the peritoneal cavity, followed by the booster doses of the antigen. And the immunization enhances the population of B lymphocytes producing antibodies specific to the antigen which we have used. So now we have to isolate such spleen cells which contain the B cells which can produce the antibodies. Now from the same animal you have to isolate the myeloma cells. So myeloma cells are the tumor cells which are having the capacity to divide and differentiate continuously, and these myeloma cells are obtained from the same immunological origin, means from the spleen itself the tumor cells are taken out. And now the second step is the cell fusion. A large number of isolated B cells are induced to fuse with the selected myeloma cells to form the hybrid cells which are called as hybridoma cells. We are using some agents for the fusion of the two cells. These agents are called as fusogens. The fusogen generally used are the polyethylene glycol, or by the electrofusion it is done, or by using the Sendai virus. So, by the use of the fusogens, the B cells and the myeloma cells are made to fuse together. As a result, there will be different types of cells formed. There may be the hybrid cells formed by the fusion of a B lymphocyte and a myeloma cell, or sometimes two lymphocytes, means a B cells, they fuse to form fused B cell, and sometimes there will be the two myeloma cells fused to form the fused myeloma cells, or sometimes there won't be any fusion. As a result, B cells will be unfused B cells produced, or sometimes myeloma cells are also unfused, and those unfused myeloma cells will also be present in the medium. So myelomas are here selected for mainly the two reasons. These cells cannot produce antibodies by themselves. These are the one which cannot produce antibodies by themselves. They contain the genetic marker HGPRT trait, permitting easy selection of the hybridomas. So here by the fusion, here we will be obtaining the hybridoma cells here, and but we have to make these myeloma cells which we are using for the fusion, those should be made, or those should be made deficient of an enzyme called as hypoxanthine guanine phosphoribosyl transferase. Next is the culture in the medium. The cells which are formed here, they are made to get cultured in the hat medium. The hat is a medium containing the hypoxanthine, aminopterin and thymidine mixture. So there will be the 5 different types of cells. There may be the fused hybrid cells, or fused B cells, or the fused myeloma cells, or sometimes unfused B cells and the unfused myeloma cells in the medium. Anyhow, when we keep all these cells in the hat medium, there the hat kills the myeloma cells. Hat kills the myeloma cells. Why? Because the myeloma cells are made deficient of the enzyme HGPRT. So the spleen cells are short-lived cells, and so they die gradually. So we are adding the aminopterin to the culture medium to kill the unfused myeloma cells in the medium. So by the addition of the aminopterin to the culture, we are making these myeloma cells which are here already deficient of the HGPRT enzyme. They cannot synthesize the nucleotides using this aminopterin. So as a result, the unfused myeloma cells in the hat medium, they die by the addition of the aminopterin. Only the hybrid cells, they survive in the hat medium, and the clone means the clone which are remaining here, means the hybridoma cells which are here, they are made to clone by making one cell, means by placing one cell in a well and allow them each cell to grow into the clone of cells. So a clone of hybridoma cells are obtained by placing one cell per well here. And the next is screening and the culture. So screening and culture is done. So for the screening, you are going to assay for the desired antibody in the culture supernatant either by the ELISA test or RIA test or the Western blot test. So RIA, ELISA or the Western blot are the three techniques, or one of the three techniques are used for the assay of the desired antibody in the culture supernatant. After this assay, grow the chosen clone of cells in the tissue culture indefinitely. So later, the specific hybridomas are either cultured in vitro, they are cultured either in vitro or passed into the peritoneal cavity to obtain the monoclonal antibodies. So later, harvest these monoclonal antibodies from the culture supernatant. So there are advantages and some of the disadvantages in the hybridoma technology. First, let us see the advantages of the hybridoma technology. So by this technology, the production of a specific antibodies to impure or the mixed immunogens is possible. It is a potential method to generate large quantities of antibodies under precisely controlled conditions. It is in vivo and the in vitro production of the monoclonal antibodies is high here. The rate of production of the in vivo production and the in vitro production of the monoclonal antibodies is high. And these are the immortal cell lines. Now, next is the disadvantages of the hybridoma technology. In this, they need high experienced qualified persons, and there is high risk of contamination also. Some antibodies are very liable, and so the activity may be lost on freezing and thawing or long-term storage. The frequent need to purify a monoclonal antibody from monoclonal culture medium is required. And now let us see the applications of monoclonal antibodies. So it is used to identify the infectious diseases and to treat traditionally untreatable viral diseases, and to identify the specific cells or the molecules in an organism, or it is used for the tissue typing or the blood typing also. It is a technology used to solve the problem of organ rejection in the organ transplanted patients. In a cancer treatment, to identify the tumor metastasis. In the follow-up therapy of the leukemia and the lymphomas, the monoclonal antibodies are best used. To measure the protein or the drug levels in the serum, and to detect the pregnancy very early after the conception here. So it is also used for diagnosing the hepatitis, influenza, herpes, streptococcus and chlamydia, and is used for identifying and quantifying the hormones. So at last, let us see the summary here. First, you have to immunize the animal. Later, isolating the spleen cells, which are containing the B lymphocyte cells, and isolate the myeloma cells, which are engineered to be deficient of HGPRT enzyme, and fuse the spleen with the myeloma cells using the PEG or the Sendai virus, etc. And culture in the hat medium. In the hat kills the myeloma cells. Spleen cells are also short-lived, so they die gradually. Only the hybridomas survive, and clone remaining hybridoma cells, placing one cell per well and allow each cell to grow into a clone of cells. Screen the supernatant of each clone for the desired antibody by using ELISA or RIA or the Western blotting technique, and grow the chosen clone of cells in a tissue culture indefinitely, and harvest antibody from the culture supernatant. Hope you have understood the topic, and please share, like and subscribe my channel. Thank you.